This video is B3 in a nutshell. It's basically me talking through the flashcards for the whole topic, B3, infection and response. And we start off talking about communicable diseases. These are the diseases that we can communicate, we can pass on to other people. So they are our infectious diseases, the contagious diseases, if you will. So we have in the past called pathogens, microorganisms, or microbes, but I want to call them pathogens. And there's 4 categories of pathogens we need to know about: bacteria, virus, protist, and fungi. And what all of these do, or certainly what the bacteria and the viruses do when they get into our body, is they reproduce rapidly, they produce toxins that make us feel ill, and they cause an immune response. Okay, the viruses actually reproduce inside our cells. This is why we can't use antibiotics to treat them, because antibiotics can kill the bacteria that float in the blood. Viruses hiding and reproducing inside the body cells means that antibiotics can't get to them. So how do pathogens cause illness? We've said about those toxins already. So they are harmful substances that poison the tissues of the body and need to be removed from the body. We've got reproduction, so this rapid reproduction, bacteria doubling around about every 20 minutes and cause an immune response. Okay, this is where the white blood cells are going to get involved. Okay, this is if we've got maybe a cut that's been infected, it will become swollen, warm, and there'll be increased blood flow to that area. And there's lots of different ways that the pathogens are spread: through food and water, insect bites, through the air as airborne droplets, direct and indirect contact. Indirect being we touch something that someone else has touched and then become infected. Now there's 7 different diseases that the exam board want us to talk about. We're going to start off with those viral diseases. We need to know things like symptoms, prevention, and also treatment. And prevention and treatment are obviously different things. Okay, if we have a vaccine, then it prevents us from getting it. It's not a treatment if we do get it. So we'll start with the measles virus. Red rash and fever can be fatal, so that means it can kill people, and young children are vaccinated against it. It's spread through droplets in the air. Tobacco mosaic virus is a plant virus. Okay, it's not something that's going to affect humans, even if we eat a plant that has been infected with it. Okay, and it creates this kind of mottled mosaic appearance on the surface of the leaves of the plants, and because it reduces the amount of green leaf available for photosynthesis, there is a reduced rate of photosynthesis and therefore reduced growth of the plant. Very easily spread, can affect all of the tomato plants in an area, so farmers would want to take good care of their tomatoes and check for any tobacco mosaic virus. Can be passed on from one plant to the other just by the farmer touching the plant and then moving it to the next plant. It can't be prevented. Okay, so there's nothing we can do to stop the plants getting it. Maybe an enclosed environment like a greenhouse could help the situation. No real treatment either, apart from to remove the affected leaves or destroy the entire plant if it's completely affected by tobacco mosaic virus. Okay, we move on to the final virus that we need to know about. This is HIV. We do not need to know that it is the human immunodeficiency virus. We can just say HIV. And it initially causes flu-like symptoms, and we take antiretroviral drugs. Okay, so they're against the virus, antiviral, and they're retro, so antiretroviral drugs. And I tend to think of it as when AIDS and HIV first came about, it was kind of more of an impact in the past. So now if people have it, there's actually 2 people who have already been declared that they don't show any signs of having HIV because they've got such small amounts of it in their body now, where they've had really good treatments. and there are pregnant women who can have a child and it doesn't pass on to the child due to the medication that we have today. So I tend to think of it as it was a real problem in the past. That's why we've got that word retro. Like if you had retro trainers, they're kind of old school, old fashioned. So antiretroviral drugs, it might help you remember that. And it basically, what those drugs do is they reduce how quickly the HIV attacks the immune system. And because it's attacking the immune system, it basically means that your body's immune system will be weakened and you can then die from another kind of infection that your body is unable to fight, for example, cancer. Okay, so which more quickly than if you didn't have HIV. So late stage HIV is known as AIDS, and that's where the immune system really is quite damaged. So we go on to the bacterial diseases. We've got Salmonella first, which is food poisoning from unhygienic conditions. And this doesn't just mean like a dirty kitchen. This means we've chopped up some raw chicken on a chopping board and then we've used the same chopping board to maybe prepare a salad. Okay, so that's what they mean by unhygienic. In this country, we vaccinate our poultry, so vaccinate the chickens to reduce the spread of the salmonella. And so if we're going to get food poisoning, it's probably going to be in another country where maybe they don't vaccinate the chicken, or when we see people who maybe go to like an all-inclusive restaurant or all-inclusive hotel maybe, and the chicken's been sat out all day on the buffet or something like that. It's giving it an opportunity for the bacteria to multiply on that food in those warm conditions. We've got symptoms of fever, cramps, vomiting, diarrhea, and these are all caused because the bacteria are releasing those toxins that we talked about earlier. Next bacterial disease is gonorrhea. This is a sexually transmitted disease. You need to know both symptoms of the thick yellow or green discharge from the vagina or penis and pain when urinating. You need both of those. I've seen already in an exam question, both were needed for the answer. Caused by bacteria, as we can see because we're talking about a bacterial disease, spread by sexual contact, treated using antibiotics, but can be prevented by using a barrier method of contraception, for example, a condom. So we go on to a fungal disease now. This one we're looking at is another plant one called rose black spot. Mostly affects the rose plants, and you can see from the picture there you've got this spots of black on the leaves, again doing exactly the same as the tobacco mosaic virus, where it's reducing the growth because the rate of photosynthesis has been reduced because we've got less areas of the leaf there that can actually undergo photosynthesis. Good thing about this one though is it can be treated. A fungicide, which is a spray that kills the fungus, can be sprayed onto the plants, or removing and destroying any leaves, but spraying it would actually help to treat it, unlike the tobacco mosaic virus. Spread by wind or water, so it can spread probably quite quickly through the plants. So next we're looking at the Protist. It's the only one we've got that is the Protist, and it's malaria. Malaria is transported using a vector, which is the mosquito. The mosquito doesn't suffer from malaria, and the mosquito is not the Protist. It carries the Protist. So the mosquito feeds on an individual's blood and then transports that. If that individual had the Protist malaria inside them, it transports it to the next person that it then feeds from. Can be fatal, causing fever, sticky blood, as we saw in the video that we watched, that is on the B3 YouTube list, so you can have a look at that. So what can we do? Well, to prevent it, we could try and get rid of the mosquitoes. Okay, so the best way of doing that is preventing them from breeding. Now they really like kind of stagnant water. That means like ponds and things where the water doesn't move that far, it's just in one place. So if we can get rid of those breeding grounds, get rid of the ponds and things where they like to breed, then it can reduce the number of mosquitoes. We can always use mosquito nets, insect repellent, put long clothes on in the evenings when the mosquitoes are out and about more, to prevent ourselves from being bitten, and also using anti-malarial medicines. And education, so it could be educating people in places where malaria is present and saying, you know, you could wear long clothes in the evening and it will help prevent you from being bitten. But it can also be if you're thinking of going on a holiday and you book in an appointment with the nurse at the doctor's and they educate you on what kind of diseases are present in the area that you're going to and how to protect yourself from those diseases. So there's lots of different ways that education can occur. Let's look at human defence systems then. So we've got the physical barriers and the chemical barriers. Physical being anything that stops the pathogens from getting in, things like saliva and tears have antiseptic properties. Sebum that comes off the skin is also good for that. We've got scabs, we've got the stomach acid, the hydrochloric acid in the stomach that kills bacteria, and we've got our other preventions like earwax and hair and things like that. Then we've got our defence mechanisms, and they kind of sometimes talk about the first, second and third lines of defence. First is the physical and the chemical barriers. Let's try and stop the pathogens getting in. Second is the engulfing of bacteria using white blood cells. More about that in a bit. And the third line of defence is those antibodies. Now in the trachea, which is the windpipe, we've got two types of cells. We've got the goblet cells and the ciliated cells. And the goblet cells produce mucus, that's basically snot, which traps the dust and bacteria that goes into our throat. And then the ciliated cells waft that mucus up to the mouth and up to the nose, so it doesn't go down into the lungs, to keep the lungs nice and clear. Okay, obviously smoking would damage those cilia, and that means that the mucus then ends up in the lungs, which is obviously not good news. Popular one, the exam board to talk about those particular cells. Viruses, as I said before, they reproduce inside the cells. So here is our cell, and the virus has put some of its genetic information into that cell where it's then reproducing, and then it destroys the cell as it escapes from the cell. And sometimes viruses can actually remain dormant. This means they just sit inside the cell for a number of years without actually having any impact on the body at all, and then they can explode out of those cells and infect the body. Okay, so that is something that can happen. Again, this is the reason why antibiotics can't be used to treat viruses, because they're inside the cells. The antibiotics would never get to them. So we've got our white blood cells, and they've got three methods of attack. Okay, first I've been talking about these toxins produced by the bacteria. We're on the top row at the moment. Okay, and those pathogens release toxins. They're going to make us feel unwell, and the white blood cell doesn't actually directly get rid of the bacteria at this point. It's just working on getting rid of the toxins by producing antitoxins, and those antitoxins, when they come in contact with a toxin, they counteract it. Okay, that's the word that they seem to like on the mark schemes. They counteract it, so it reduces the amount of toxin going through the blood and therefore reduces how we feel. Okay, the next one we're going to look at is the middle row, and that is the engulfing and digesting of pathogens. Okay, and we call this, these particular types of white blood cells, the phagocytes, okay, or the phagocytes, depending on how you say it. And they literally circle around the pathogen, engulf it. They've got enzymes inside which digest it. Some of those parts of the pathogen will be used by the white blood cell, and some are excreted. Okay, so that's the engulfing, using that process of phagocytosis or phagocytosis, depending on how you want to say it. And then we've got the releasing of antibodies. Now, antibodies are a bit like the whole lock and key business of enzymes, but we don't say lock and key. We just say they have a specific shape. So you can see around the pathogen there are sections of protein called antigens, and these are like the markers. They say, hello, I've come from somebody else or somewhere else and I am foreign to your body, therefore instructing the white blood cells to fight it. And same reason why we talked about it in B2, our body wants to reject any new organs that are put into it because of these antigens. So what happens is the white blood cells produce these antibodies that then latch on and attach to those antigens, and once they've attached on all the way around, then the pathogen becomes inactive, okay, and they are a specific shape to the antigen. So as I said, we've got those specific shapes, and there's some examples there on the left. Okay, TB, so tuberculosis bacteria have a number of adaptations that enable them to resist ingestion. This is just kind of an interest bit really. They produce chemicals that prevent white blood cells from being attracted to the sites of infection. They also have a waxy cell wall that resists enzyme attack and secretes chemicals that block its destruction. So just saying how kind of bacteria have evolved to become resistant to demolition really. So antibody levels during infection. Now this first infection could be being infected, or it could also be when you have your vaccination. So when you first are infected, either by picking up that disease from somebody or by having a vaccination, it encourages the white blood cells to start making the correct antibodies. And we can see that in this first peak on the graph. So this is the initial white blood cells, those lymphocytes that we call white blood cells, first understanding how to make the correct antibody. And then the pathogen, after it's been destroyed, doesn't need those antibodies to stay around. However, the lymphocytes, the white blood cells able to make the correct antibodies, are still in the blood, and we call these memory cells. So when we get our second infection, we rapidly, now that's really important, we rapidly make the correct antibodies, so we can fight something without even feeling any of the symptoms. Okay, so when we have our vaccine, when we then are actually infected, technically we're contagious and we can pass it on to other people, but we won't even feel ill. Our body will be making antibodies using those memory cells. Okay, so they're white blood cells that we call lymphocytes, and they're also memory cells. They've got the memory, the ability to make the correct antibody because they've met this disease before. Okay, and that rapid increase in antibodies means that we fight it without feeling any of the impact. Again, those antibody levels fade off again because once we've recovered from it, we don't need the antibodies to stay in the blood, but we do need the memory cells to stay there in case we get infected again. Active immunity can be either getting ill in the first place and our body doing what we've just talked about and making the antibodies, or it can be being infected by a vaccine, small amount of dead or weakened pathogen that stimulates the production of antibodies and memory cells. Okay, we are actively doing this. So we are taking the part of the disease or the part of the antigen that's in the vaccine and learning how to fight it actively ourselves. If we have lots of people become immune to the pathogen, either they've had it or they have had the vaccine, and there's some people who haven't been affected, then we call that herd immunity. Because there's less people to spread the disease, the few remaining people who haven't had the vaccine or haven't been infected are protected. Okay, so we call that one herd immunity. And then we've got passive immunity. This is the one where we're not able to make the antibodies at all or quickly enough in order to fight the disease. And the classic example of this is a snake bite. And with a snake bite, we're not able to make the antibodies quickly enough in enough quantity in order to help us survive. So what's done is the snake toxin is put into a horse, at a non-lethal dose. So it's not going to kill the horse, but because the horse is so big, it's able to make lots of antibodies, and those antibodies can then be extracted from the horse and given directly to the person who's had the snake bite. Okay, we call this passive immunity because somebody else has made those antibodies for us. So it's not giving the white blood cells to the person who's had the snake bite, it's actually giving the antibodies directly from the horse, okay? And that's obviously done and put in storage ready for a snake bite. They don't infect a horse as soon as someone's been bitten by a snake. They've done this previously, ready for that situation. So vaccinations, talked a little bit about these already. A person is injected with a small amount of dead or inactive pathogen. The white blood cells react to the dead pathogen, so they produce those antibodies, learn how to fight it, making memory cells. And the person, if they then become infected with the pathogen without even knowing what's going on, the memory cells will recognise the pathogen and produce antibodies, and the person won't even feel ill. Okay, it is destroyed, and it's happening to us probably right now for different diseases, and we don't even know it's happening, but it's because we've got our immunity. So benefits of vaccination is there's less chance of getting infectious diseases, herd immunity can wipe out diseases, for example polio, and millions of pounds can be saved on treatment because we're using preventative measures. However, there are a small chance of harmful or permanent side effects. So why do we have a new flu vaccine every year? Well, annually, the pathogen that infects us with flu mutates. It creates a new strain. Okay, we're hearing the word variant a lot at the moment. And the new strain of the disease is resistant to the vaccine that we've already had if we're weak or vulnerable and we've had that flu vaccine. So because it's mutated and created a new strain that nobody is resistant to, it will spread really quickly. Okay, there's no herd immunity. So what we do is each year we create a new vaccine to be given to those vulnerable people to protect themselves against the new strain of the flu virus. So let's go through some other key points that we haven't talked about yet. Painkillers are just literally there to do what they say, relieve the pain, kill the pain. They relieve symptoms of the infection, but they don't actually kill the pathogen, so they just make us feel a bit better, maybe take down a fever, that kind of thing. I've talked already about how antibiotics kill infectious bacteria, but they don't destroy the viruses because they reproduce inside cells. Antibiotics has greatly reduced the number of deaths from bacterial diseases, no surprises there. And again, they're specific, so a bit like those antibodies, the antibiotics are specific and different antibiotics are used to treat different bacterial infections. We've then got this business about overuse and inappropriate use. I'm going to talk about inappropriate use first of all. Remember, antibiotics must be prescribed by the doctor. So we can't just get antibiotics in the shop, we have to get them from the doctor. Now, inappropriate use is if the doctor says, right, you're going to take all of these antibiotics over the next 7 days, and you get to day 4 and you think, actually, I feel all right now. And that's because the antibiotics that you've had have had a really good opportunity to kill lots of the bacteria in your body. And we can cope with this low level of bacteria in the body. That means that we don't feel ill. So we stop taking the antibiotics, and then the bacteria has a chance to multiply again and we get ill again. So inappropriate use is the not taking the entire course of antibiotics. And then we go on to overuse. And the overuse of antibiotics is if you keep going to the doctors and the doctor keeps giving you antibiotics every time you're ill. And what happens is some of those bacteria will have mutated and formed their own small population. And if they are not killed by the antibiotics, but all the other bacteria are, they now have a greater chance of survival. They're going to get to all the resources because all the other bacteria have been killed, and then that population of the new strain will reproduce rapidly. So this is why we shouldn't have antibiotics when we can allow our body to fight things itself. Okay. We need to know some of the drugs that we can get from plants. So there's a heart drug called digitalis. Think of your digits. It comes from foxgloves, and gloves are used to put over our digits. So if you think of digitalis and your digits, your fingers, and then we put a glove on that, you come up with foxglove. Painkiller aspirin originates from willow. There are 2 L's in painkiller and 2 L's in willow, so that's useful to try and help us remember it. And then penicillin comes from the penicillium mold. Okay, so that one's easy. And nowadays, because we've got so many people on the planet, we synthesize drugs, so we artificially make those drugs by pharmaceutical companies. So we've got enough for everybody who needs them. Now we're going to talk about drug trials next. When we go into a drug trial, if the drug has been made by a particular company and they're going to do the drug testing, then they want to remove bias. They don't want to try and convince the volunteers that they have recovered better by having the drug. So some people will have the drug, some people will have a fake drug called a placebo, which looks like the drug but has no effect. It could be a sugar tablet or a sugar injection. It's used for a comparison, but the fact that the volunteers and the people doing the drug trial don't know who gets the placebo and who gets the drug until the end of the drug trial, we call it a double-blind trial. So that means nobody knows. Double because the doctors don't know and the patients don't know, or the volunteers don't know. If it was being tested by an independent company who were not part of the construction of the drug and they're just going to get paid regardless, then they could know who's getting the placebo and who's getting the drug. So that would just be called a blind trial. And that one, the patients still wouldn't know who got the drug and who got the placebo. And then the third alternative is an open trial where it's just everybody knows they've got the drug and we're just seeing, is that drug going to do the job? There's 3 reasons why we do drug testing. The first one is efficacy. Does the drug do the job? Does it actually stop the disease? Dosage. What is the smallest amount of the drug we need to give to people to actually treat the condition? This has obviously got health benefits. We don't want to have lots of a drug if we don't need as much of it. It could cause some side effects, and also a financial implication of only needing a certain amount of a drug rather than taking too much. And also side effects, toxicity, how does the body react to that drug? So those are the 3 reasons why drug testing happens. And there's really kind of 4 main stages to drug development. We're going to start up here with the preclinical development. This is the stuff that isn't involving humans, okay? So this is testing on human cells, testing on animals, and looking at computer models. What do we already know about the drug? And that's called preclinical development. You can see on average it takes about 3 years for that stage. Then we've got the clinical development, which is roughly put into these 3 stages or phases. But again, this can change depending on what the drug is and how it's being developed. So the clinical development is where human testing begins. And it starts off in phase 1 where we've just got volunteers. So these people are not ill, but they volunteered to see how the drug affects them. And this is looking at side effects. So we can't see, is it going to cure them because they're not ill, but we're looking to see what the side effects are. Phase 2 of the clinical development is where we test on patients, and this is to find out about dosage. How much do we need in order to recover? And also looking at the minimum side effects that occur when we do that. Phase 3 is rolled out in large numbers now, lots of people, and we're comparing the drug to the placebo. Okay, that's for our comparison. It then goes to the regulatory review. This, you'll know a lot about this from the news lately, because this is where it goes off to check that the drug trial was done in the right way and that everything was done correctly, and more importantly to check that the benefits of the drug outweigh the risks. And then the drug is launched, okay? And it's still monitored and developed over time to check that it's still doing the job it should do and that there are few side effects. So we talked about the open drug trial. This is where everybody knows that it's just we're taking the drug. The blind trial, where only the doctor knows who's taking it, and that's because it's an independent company that's testing it, not the company that designed the drug. And then the double-blind trial, which is the one we talk about most frequently, where neither the doctor or the patient knows who has had the drug and who's had the placebo. I think we understand that now. Moving on to some triple content, then we're looking at monoclonal antibodies. Now, monoclonal antibodies are the ability to have lots of antibodies that are all the same. Once a white blood cell starts producing antibodies, it can no longer multiply. So we've got a fixed number of white blood cells that can make a particular antibody. That's point number one. Point number two is that white blood cells, when they start making a particular antibody, they're a one-trick pony. They can only make one type of antibody. So what if we wanted to make a cell that can make the antibody we're looking for and reproduce? So that way we can get loads of those antibodies. We can use them for various different things that we'll talk about, but we can also use them for passive immunity, the snake bite scenario, or any other time where we want passive immunity. So here's what we do. We take effectively our vaccine, which contains the weak pathogen, the antigen, and we infect an animal, in this case a mouse. That will then start producing the antibody for that particular antigen. And we've removed those, we're going to call them lymphocytes now, the white blood cells that are making that particular antibody, from the blood of the mouse. We're then going to take a tumour cell, so this is a cancerous cell. We've removed the nucleus from this because we're not looking for that genetic information, we're just looking for a cell that can reproduce. And we fuse together that antibody forming cell with the tumour cell, and because it's got features of both, we call it a hybrid. Okay, like a hybrid car or a hybrid bike, it's a hybrid, but we're actually going to call it a hybridoma. Okay, so we've made some hybridoma cells, and they are now able to reproduce and produce lots of antibodies. Okay, they've cloned from one particular cell, so we call them monoclonal antibodies. Okay, there's the information. You can pause that if you need to. Okay, but I've talked through all of that. So how they're used, they can be used in pregnancy test kits. More about this when we talked about it in class. There is a hormone called HCG that women produce when they are pregnant, and monoclonal antibodies can be used to detect that hormone in the blood, in the urine, and inform if the woman is pregnant or not. Can also be used to locate blood clots. Can be used to detect lots of different chemicals in the blood, in blood testing kits. A lot of disadvantages from monoclonal antibodies at the moment, so side effects, they need to be worked on. It is a new technology thing, okay? They can be used to diagnose and treat cancer, where they can have a particular shape to them that can bind onto a cancerous cell but not onto a normal body cell. We can attach a radioactive isotope to the end of the monoclonal antibody. We talked about these isotopes in P4, that's the physics topic. So that attaches to one end of the monoclonal antibody, and then the other end attaches to the cancerous cell. So it means that when that chemotherapy is put into the blood and it flows through the blood, rather than just killing lots of different cells, it will bind to the cancerous cells and localize that radioactive treatment to those particular cells, because it won't bind to the normal body cells. Okay, so that's a really good advantage. Means we can use less of the drug, less side effects. We've got this weird bit about plants on the end of the topic, not really that difficult, but they have put it as triple science. So if our plant has a disease, what might we notice? It could have stunted growth, spots on the leaves, growths, discoloration, the presence of pests. How could we identify the plant disease that the plant has got? We could look it up on a website, we could look in a gardening manual, we could take it to a lab where it's identified, and there are monoclonal antibody testing kits that can be used as well. There are a couple of deficiency conditions we need to know about. One is if the plant has a lack of nitrate. Now, nitrogen is found in amino acids. Amino acids build up to make proteins. So if the plant is lacking nitrogen in the form of nitrate, which comes from the soil, then it's not going to have very good growth because it can't build those amino acids and therefore proteins, which we know are for growth and repair. The plant may be suffering from yellow leaves. This is a lack of magnesium, which creates the chlorophyll, and we call this chlorosis of the plant. So it doesn't have enough magnesium to make the chlorophyll needed for photosynthesis. Plant defences, so this is a defence against microorganisms, so bacteria, pathogens, the viruses, the fungi. The cellulose cell wall restricts anything from going inside the cell. On a larger scale, the waxy cuticle on the top of the leaf prevents pathogens from getting into the leaf. And then on an even larger scale, the bark on trees is dead cells which fall off, and then the bacteria or other pathogens fall off with it. Chemical responses, antibacterial chemicals or poisons that deter herbivores, so these could be insects or larger herbivores that are feeding off the plant. And then if we look on a larger scale, not just looking at microorganisms, but looking at animals, we've got thorns and hairs to deter animals. We've got leaves which droop or curl when they're touched, so they maybe look more pointy than they are, or they're disguised, and mimicry to trick animals, so mimicking other plants that animals know are already poisonous to them or not very pleasant to eat. So this one tells us a bit more about the pregnancy test kit. I'm not going to go into it now. I'll go into that one another time in class. And we've got some possible longer answer questions here that you could have a go at, so you could pause this and try these questions or look at the flashcards. Hope that you found that useful. Thank you very much.